Assalamu alaikum. Welcome to my YouTube channel. Today I am here to explain you about kidneys and nephrons. Uh, in this lecture we will understand the basic structure of kidney and the nephrons, which are the functional unit of kidneys. Before we start the lecture, let me remind you that please subscribe my channel and share it with other students. So let's start. Kidneys are the excretory organs of in our body, and these excretory organs, kidneys, they remove urea, which is the metabolic waste product. What is excretion? Excretion is the removal of metabolic waste products from our body. These metabolic waste products are harmful and toxic, and they must be removed from the body. For example, urea and carbon dioxide. So kidneys remove urea in the form of urine. So this is the basic structure of kidney. So kidney has two major regions. One is called cortex and the other is called medulla, or more correctly we can call it renal cortex. Renal, R-E, let's say if I write its complete name, renal cortex and renal medulla and renal pelvis and renal artery and renal vein. Remember, the term renal is associated with kidney. So renal cortex is the outer region of the kidney, and the inner region is called medulla or renal medulla. Cortex, in the cortex region we can see the small dots. These small dots are actually nephrons, which are the filtration plants of the kidney, where the filtration takes place. And the inner portion, the inner region of kidney is called medulla. Here in this portion of medulla there are pyramid-like structures, pyramids, and these pyramids contain collecting tubules which collect the urine, and then they pour it in this filter-like structure, a funnel-like structure. This funnel-like structure is called renal pelvis. The filtered urine is collected through collecting duct and then eventually is poured in this funnel-like structure called renal pelvis. And this renal pelvis, then after collecting the urine, then it adopts the shape of ureter, and this ureter then takes the urine towards the urinary bladder, where the urine is stored temporarily for some time, and then from there the urine is discharged out of the body. Okay, now let's talk about nephron. What is nephron? It is, as I told you, here the filtration takes place, and from the blood the urea is filtered in the form of urine along with certain amount of water and certain amount of salts. So this nephron, it has a cup-shaped structure, a cup-like structure like this. This cup-shaped structure is called Bowman's capsule. It is named on the name of its scientist who discovered it, Bowman. So Bowman capsule is like this, and it is inside this Bowman capsule we can see the network of blood capillaries. This network of blood capillaries is called glomerulus. And how this glomerulus is formed? Here we can see an artery which I draw with red color. This artery divides into a small arteriole. Remember, the short form of artery is arteriole, like book, booklet. Similarly, artery, arteriole. So this arteriole is called afferent arteriole. Its name starts with A, afferent arteriole. And when this afferent arteriole enters into this Bowman capsule, it divides and subdivides and it forms a network of blood capillaries which is called glomerulus. And when this glomerulus, from this glomerulus, another arteriole comes out, bringing the blood out of this glomerulus. This arteriole is called efferent arteriole. Now there are two terms and there are two types of arteriole. One is afferent arteriole, afferent A, and the other is efferent arteriole. So afferent arteriole is bringing the blood towards the glomerulus, inside the glomerulus, and efferent arteriole is bringing the blood outside of the glomerulus. Now there is a difference in the diameter of these two arterioles. If you can see, the diameter of afferent arteriole is broader, is larger, and the diameter of this efferent arteriole is comparatively smaller, narrower. Why there is a difference of diameter? And the whole mechanism of nephron is based on this difference of the diameter. This is the mechanism how the filtration of nephron inside the nephron and how the filtration inside the kidneys it takes place, how it works. This is the whole science is behind is working on the basis of the difference of this and this diameter, the difference of the diameter. Now, as I told you that here afferent arteriole is broader, so it is bringing more blood into the glomerulus, and the efferent arteriole is narrower, so it is bringing lesser blood, it is bringing back lesser blood, and due to the difference of this diameter, more blood is entering into the glomerulus and a high blood pressure is developed inside the glomerulus. So because of this difference, now more blood is entering into is flowing inside the glomerulus and lesser blood is coming out. So a high blood pressure is developed inside the glomerulus. And due to this high blood pressure, the smaller molecules present in the blood, they come out, they are filtered. And what are those smaller molecules? These smaller molecules are molecules of water, and these smaller molecules are molecules of urea, and these smaller molecules are molecules of glucose and amino acids and salts like salts of potassium and sodium and bicarbonates, etc. So all the small molecules which are present in the blood, under high blood pressure, they are filtered off, and as the walls of the blood capillaries and as the walls of the Bowman capsule, they are partially permeable membrane, they are semi-permeable membrane. So here we can see that there are small pores, so the blood will come out, certain part of the blood, I should say, the smaller molecules like water and glucose and urea and amino acids and excessive salts, all of them, they will come out under high pressure. They are squeezed out. So, but remember that not the large molecules, they do not come out of the partially permeable membrane, because this is the principle of you can say partially, this is the mechanism of partially permeable membrane. So the large molecules of blood like proteins, the molecules of proteins, what kind of proteins are present in the blood? For instance, globulin, albumin, fibrinogen, etc. They do not come out of they cannot cross the partially permeable membrane of the glomerulus and the Bowman capsule. So they will not come out. Similarly, the red blood cells, white blood cells, platelets, etc., they will not come out of the partially permeable membrane because they are large. So they will stay inside the blood. But all the other small molecules, including water and glucose and amino acids and urea, they come out of the blood and they will start traveling in this coiled tube, convoluted tube. This is called convoluted tubule. Why it is called convoluted tubule? Because it is coiled. Convoluted means coiled, right? So not straight. This coiled convoluted tubule is taking the filtrate from this region, and remember that this region is present in the cortex. This region is present in the cortex, here in the cortex of the kidney, while this region is present in the medulla, the loop of Henle and the collecting tubule. They are present in the medulla, renal medulla. So here we can see that when the filtrate is passing through the convoluted tubule, so this part of the convoluted tubule is also called proximal. It means that beginning, the tubule which is present in the beginning, proximal convoluted tubule, and this is distal convoluted tubule. So when the filtrate is passing through the convoluted tubule, especially the proximal convoluted tubule, so you can see that the blood capillaries are also chasing them, and also the blood capillaries are surrounding and chasing the loop of Henle. Loop of Henle is a U-shaped structure, and it is named on the scientist who discovered Friedrich Henle discovered it. So this U-shaped structure, it is like U-shaped structure, loop of Henle. It is present in the renal medulla. So the blood capillaries are surrounding it. So what is taking place in this convoluted tubule, proximal convoluted tubule and the loop of Henle? Remember, selective reabsorption is taking place in the loop of Henle and in the proximal convoluted tubule. Why selective reabsorption is taking place? Because all of the small molecules should not be removed, because all of the water present in the blood, it should not be removed. Certain amount of blood is reabsorbed back into the blood, because our body needs water, our blood needs water to maintain a certain amount of osmotic potential. All of the blood should not, all of the water should not be lost. That's why selective reabsorption should take place, and it is very necessary. Similarly, glucose, despite of the fact that glucose is a small molecule, but it should not be removed. It is very useful substance. It is providing energy. So glucose must be reabsorbed. So selective reabsorption is taking place here, and this is the purpose of the blood capillaries, why the blood capillaries are chasing. They are absorbing, reabsorbing certain amount of water and reabsorbing certain certain amount of salts, and they are reabsorbing all of the glucose and all of the amino acids. So because these are useful things. So selective reabsorption takes place here. First step is ultrafiltration, which takes place inside the glomerulus under high blood pressure, and the second step is selective reabsorption. So selective reabsorption takes place in this tubule and in this loop of Henle. And while the filtrate reaches up to this region, now it forms urine, which must be removed, because now urea is dissolved in water and a certain amount of salts. So all this solution, now it is called urine. Now urine formation takes place, and the third step is urine formation and urine collection. Now urine will be collected in the through the collecting duct. So collecting duct is here. Now when the nephron is ending, the duct is the tubule is ending here. So it is pouring its urine, its filtrate into the collecting duct. So another, this arrow is representing that here there will be another nephron. Here at this point there will be another nephron which is pouring its urine into the collecting duct, and here there will be another nephron which is pouring its filtrate into this collecting duct. So the purpose of the collecting duct is to collect the filtrate, that is urine, from all parts of the uh, kidney, and now they will pour it into this funnel like structure which is called renal pelvis, and renal pelvis will send this filtrate, this urine, through ureter into the urinary bladder. And remember that the one kidney, it contains almost 1 million nephrons. Now at this point I would like to explain this glomerulus, that how this inside this glomerulus ultrafiltration is taking place under high blood pressure. To simplify and to further elaborate, I will give you an example of water pipe. Suppose consider a water pipe, and in this water pipe water is flowing under high pressure, and but this pipe is leaked and it has small holes or small pores, so water will be coming out because of the high pressure in the form of spray through these pores and through these holes. You might have observed sometime water pipe, a leaked water pipe passing through a lawn or from any other point. So so the water is coming in the form of spray. Why? Because inside the water pipe the pressure is very high, and due to the small pores the water is coming out, spraying out. Similarly, under high pressure, through these small pores of the partially permeable membrane, the small molecules of water and urea and other substances, they are coming out. The same phenomena is taking place here. So here we can understand the structure of kidney and nephron. I hope that you have understood and enjoyed my lecture. Thank you very much.